Good evening, everyone, and welcome to tonight's program, Demystifying COVID-19, the SARS-CoV-2. My name is Susan Van Epps, and I'm the Division Manager for Programming and Community Engagement at Loudoun County Public Library. Tonight's program is the first in a three-part series co-hosted between the library and the HHMI Janelia Research Campus. On behalf of the library, I want to extend our sincere thanks to the entire Janelia team for their ongoing program partnership with us, for their work organizing this very important series, and for graciously sharing with all of us their world-renowned network of scientists. And now I'd like to introduce to you Mr. Ron Vail, HHMI Vice President and Executive Director of Janelia Research Campus in Ashburn, Virginia. Good evening, Ron.
Thank you, Susan, and it's great to do this series with you. As Susan said, Janelia is a life science research campus in Ashburn, Virginia, and we're part of the Howard Hughes Medical Institute. And I'd like to also welcome everyone to Dialogues, which is the name of this series. And normally, Dialogues is a series where we invite the public to come to our campus and hear a great science lecture and also interact with our scientists. But this year, due to COVID-19, that kind of public engagement in person on our campus is not possible. So we're continuing in this remote format. And also it's an opportunity for you to learn about the science of COVID-19, which is obviously science that's on everyone's minds right now. And with that, I'd like to introduce the organizers of this series, Sarada Viswanathan and Janine Stevens. And now I'll turn it over to Janine.
Thank you, Ron. Hi, everyone. I just want to mention a few quick housekeeping notes before we get started with the fun stuff. I want to let you know that tonight's talk will be roughly 40 or 45 minutes in duration. And afterwards, we will gladly take audience questions. For those of you who are not familiar with using Zoom, you should see either a raise hand button and or a Q&A button at the bottom of your screen. So you can either write your question in the Q&A box and we will read it to the speaker after the seminar, or you can click the raise your hand button and we will call on you to ask the question yourself, but you will have to make sure that you're not on mute. Please keep in mind that this is a three-part series taking place over the next few weeks, covering everything from the basic biology and immunology of SARS-CoV-2 to how it spreads and efforts to treat and prevent it. So if you don't get all of your questions answered tonight, you will definitely have other opportunities. And with that, I want to turn it over to my colleague Sarada to introduce tonight's speaker.
Thank you, Susan, Ron, and Janine, and thanks to everyone for joining us this evening. Today, we are very pleased to have with us Dr. Angela Rasmussen, an Associate Research Scientist at Columbia University's Mailman School of Public Health. She is also a member of the World Health Organization's ad hoc expert group on preclinical models of COVID-19 disease. She holds a PhD in microbiology from Columbia University and has previously worked as a researcher at the University of Washington. A bit about her work. Dr. Rasmussen is a virologist who studies host response to infection. She is particularly interested in viruses that are highly pathogenic and has worked on a range of viruses that pose a major threat to global public health, such as Ebola, dengue, influenza, and now SARS. In this three-part series, Dr. Rasmussen will cover basics about COVID-19 pandemic, including what we know, what we don't know as yet, and what scientists are working hard to discover. And as Susan mentioned, today in the first talk, she would cover the fundamentals of SARS-CoV-2, as we know, the virus that causes COVID-19, including what we know about how it infects the cell, how the virus causes the COVID-19 disease, and how our body, our immune system, responds to this infection. So Dr. Rasmussen, on behalf of the audience and all of us here, a very big thank you for all that you do. Your research and the time that you take to spread awareness within the community, which is so important at this point in time. And I must tell you this, that your talk, your papers, your tweets have been my go-to resource in trying to understand this virus, this disease, and the situation in general. So once again, a very big thank you and welcome.
So thank you so much, Sarada, for that really kind introduction. And now I'm going to share my screen and get right into it. Okay, so before I start every talk, I do like to express my territorial acknowledgement and equity statement. Today I'm in Seattle, so I'm presenting from the unceded ancestral homelands of the Duwamish people. I work at Columbia University, which is located on the traditional lands of the Lenape people, and I acknowledge and honor the first people of these territories and their tribal governments, their histories and their ancestry, and their roles to this day that in caring for these lands. And I'd also like, before I get started, to acknowledge that there's a history of systemic inequity in academic science that spans decades, and in the case of Columbia University, centuries. Columbia University was founded using the profits from the transatlantic slave trade and has excluded women and people of color from the academic community for nearly 2 centuries. This has left a long and painful legacy of racial and gender-based inequality that continues to this day. And I encourage all of you watching this presentation, especially in the public, to consider how you can contribute to making not only medicine and science, but our larger society a more equitable place for everybody to exist in and to do this type of work. So thank you so much for having me here again. It's really an honor to be able to talk to you today about SARS coronavirus 2 virology. And first, I'm going to start off by just kind of taking you through the steps that we all went through at the beginning of the year, trying to understand what was going on from a virologist perspective. I think one thing that has kind of gotten lost in this pandemic is the fact that scientists have learned about this virus really at the same time that the rest of the world has. This isn't something that we've all been working on. You know, nobody's been paying attention and the virus came out. We all learned about it around the same time that the rest of the world outside China did. And that was through this email service called ProMed Mail, which is a website that posts incidents of different types of infectious disease outbreaks. And so this was the ProMed post that went up on December 30th, 2019 about undiagnosed pneumonia in Hubei province, China, and a request for information for any scientists who might have more information or be able to help figure out what was causing this. And everybody, you know, from a virologist perspective, we all were aware of this. I have numerous collaborators who work with scientists in China, including at the Wuhan Institute of Virology. People knew that there was this undiagnosed pneumonia. We didn't know what was causing it. So everybody was very curious to see what it was and to keep an eye on it to see if it was going to become something serious that we all needed to pay attention to, which obviously it did. And on January 10th, a week and a half later, Eddie Holmes, who is an evolutionary biologist and virologist at the University of Sydney, on behalf of his collaborators in China, posted the first genome sequence of this virus. And that was how we found out that, in fact, it was a novel coronavirus. So the entire genome was sequenced. We analyzed that compared to all the viral genomes that have been sequenced in the past and are known. And based on that analysis, we were able to determine that it was a coronavirus. More importantly, we were able to determine that it was a coronavirus we had never seen before. And at later talks in this series, I'll be talking about how this information allowed people to basically begin designing vaccines right away without actually isolating the infectious virus itself. So this was a very important finding. This is when we decided to give it the very catchy interim name of 2019 nCoV or nCoV 2019. About a month and a half later, it got a real name. And that was initially the World Health Organization, I think, was frustrated that the way that we name viruses, the International Committee for the Taxonomy of Viruses, hadn't come up with a name yet. So they decided to name the disease instead of the virus. And this is important only because I think a lot of people are confused about how viruses get named. It used to be kind of a free-for-all, and whoever discovered the virus got to name it. Then they developed this International Committee for the Taxonomy of Viruses, and now they are in charge of naming viruses, and that's kind of internationally accepted as the rule. But the World Health Organization in this case stepped in because honestly, nCoV 2019 or 2019 nCoV are pretty terrible names. And they were always intended to be interim names. So they decided on February 11th to name the disease that this virus causes. And that was COVID-19 for coronavirus disease 2019, after the year that the virus emerged. So a little bit later, the International Committee for the Taxonomy of Viruses, or on Taxonomy of Viruses, did name it, and they named it because its genome sequence showed that it was most closely related to SARS coronavirus classic. So they named it the very clever name of SARS coronavirus 2. SARS coronavirus itself did not become SARS coronavirus 1. I guess it's just SARS coronavirus original recipe. This is SARS coronavirus 2 or SARS-CoV-2. And so that's the name that has stuck. And people can debate amongst themselves whether that's a good name or not. I think it is what it is, and that's the name of this virus for now. So when and how did this virus emerge? And this is a very popular topic to speculate on. I'm not going to be getting into that. You're welcome to ask me questions at the end of this about what I think about that or the evidence about emergence. But I'm just going to cover this very briefly in terms of the events that happened for the virus to emerge. So initially, it was thought that the virus emerged at the Wuhan, Huanan, which means seafood, market. And this was a market that sold seafood, as the name suggests, also sold live animals. And this was thought to be, this might be where it emerged, because SARS coronavirus itself was shown to be amplified in palm civets, which is another type of wild animal that is sold sometimes in China in wet markets for food and for fur. And because SARS originally came from bats, but was thought to have been amplified in these palm civets, they thought perhaps one of the live animals that was being sold at the Huanan seafood market was the source of this virus, because there were a lot of cases that seemed to be associated with it. However, other studies, other cases of this undiagnosed pneumonia were tested retrospectively for SARS coronavirus 2 once a test was available. And they published in February in The Lancet that the first detected case was actually presented to the hospital on December 1st, 2019. And this case had no market exposure. So now it's thought that the virus did not emerge at the Huanan seafood market, rather that somebody was shopping at that market, and there was basically a super spreader event where a number of people were infected. Those people went on to infect others, but this virus was already spreading prior to the mass transmission event at this market. So we still don't know where this virus came from. Originally, it came from a virus that likely circulated in wild bats in China. We don't really know if there's an intermediate species involved or if it was transmitted directly from a bat to a human. But we do know that it didn't actually emerge from an animal at this market. So there's still a lot of mystery involved about that. But what we do know is a lot more about what this virus is and how it works. This virus is what's called a beta coronavirus. It's a coronavirus, as the name implies. It's in the family Coronaviridae. It's the genus beta coronaviruses. There's alpha coronaviruses, beta coronaviruses, and gamma coronavirus and delta coronaviruses. The ones that we are most interested in that have emerged recently within the last 20 years are beta coronaviruses. So SARS coronavirus classic is also a beta coronavirus in the same subgroup of the beta coronaviruses called the sarbecoviruses, which is not a very clever name either. That means SARS-like beta coronaviruses. MERS coronavirus is another virus that has emerged. It emerged in 2012. It is transmitted zoonotically to people by dromedary camels, although it is also derived from a virus that came from bats. It emerged in the Middle East, which is where its name comes from. It's Middle East Respiratory Syndrome coronavirus. It's in a different subgroup called the merbecoviruses, which, like the sarbecovirus genus, is basically MERS-like beta coronaviruses. And so that's what all these viruses are related to. There are two other beta coronaviruses that cause disease in humans. Those are common cold type coronaviruses, and there's two other alpha coronaviruses that also cause common cold-like disease. So these viruses are, they look like this. They look like crowns, which is where coronaviruses get their name. And that's in part because the surface is surrounded by a membrane or an envelope that's derived from the cell membrane that the virus buds out of when it replicates. This membrane is decorated with these large spiky proteins, which is cleverly named the spike protein. And we'll be talking about that more later in terms of immune responses to the virus. The spike protein is also very important for the virus being able to infect cells, because the spike protein is what binds the virus's cellular receptor, which is a molecule called ACE2. And this is the structure of the virus. So SARS coronavirus, all coronaviruses are what we call positive sense RNA viruses. So their genome, unlike ours, which is double-stranded DNA, is a single strand of RNA. And RNA is similar to DNA. It is less stable than DNA, but there are a number of RNA viruses. Other RNA viruses include influenza, Ebola, rhinovirus, which causes the common cold, poliovirus, measles. These are all RNA viruses. What positive sense means is that in the cell, normally, we make RNA from our DNA through a process called transcription. And those RNAs are basically messages from our DNA in the nucleus that gets sent out of the nucleus into the cytoplasm, which is the part of the cell that surrounds the nucleus. And there, those encode proteins, and those are translated. So messenger RNAs are positive sense, single-stranded RNAs that our cells make that are important for making proteins and expressing genes. So we will be talking about that in a moment, because those are important pathways that viruses hijack effectively to replicate within their host. Another interesting feature of coronaviruses is that for RNA viruses, their genomes are very large. And as I mentioned a moment ago, RNA is much less stable than DNA. So RNA virus genomes tend to be much smaller. This means that they usually have to interact with their hosts a lot more, since they can't encode every single protein they need to replicate. DNA viruses cannot either, but they are able to encode a lot more proteins than RNA viruses because their genomes are much larger. So this is what the coronavirus virus particle, or virion, looks like. It's this outside envelope with a shell inside that's called the capsid. In this case, the RNA genome is bound to a protein called the nucleoprotein, or nucleocapsid, that protects and stabilizes the RNA genome within the virus particle. And in order to infect a cell, which I'm going to go through in more detail in just a moment, the spike protein has to bind its receptor. The virus gets internalized in the cell. It spits out its genetic material into the cytoplasm, and then it begins hijacking host processes to replicate its genome, make viral proteins, and assemble new virus particles. So this is how the replication process works. When you have a coronavirus that gets into your nose, any coronavirus, but in this case, SARS-2, it will bind its cellular receptor, which is a protein on the surface of the cells in your epithelial airway tissue. That's called ACE2. ACE2 is involved normally in controlling blood pressure. It's part of a system called the angiotensin-renin axis, which controls blood pressure. It controls the way that basically your vascular system works. And I don't have any slides on it, but I'm more than happy to discuss some of the implications for ACE2. The thing is, we don't know very much about how ACE2 is affecting pathogenesis, or the process by which the virus causes disease. But there are a number of hypotheses. So even though I don't have a slide about it, I'm more than happy to discuss those at the end. Anyways, after binding ACE2, there's actually another protein. This figure is simplified. There's another protein on the surface of the cell called TMPRSS2 that has to sort of prime the spike protein to bind ACE2, and it does that by clipping off a section of it so that it exposes the receptor binding domain of the spike protein. After it binds ACE2, it gets taken up into the cell into a compartment called the endosome. And you're not going to be tested on all of these terms after this, but what's important to know is that when cells take up things from the outside environment, including viruses, they usually get put into one of these compartments. And that's one of your cell's first lines of defense, actually, against viral infection, is that these endosomes can become acidified and dissolve anything that's inside of them. And so the viruses are very clever in the way that they've evolved. They're not actually clever because they're not sentient, but they have evolved ways to take advantage of endosomal acidification. In this case, it causes conformational changes in the proteins that allow the virus to fuse and stick its RNA out of the endosome into the cytoplasm at large. And as I mentioned, the viral genome is positive sense, like a messenger RNA. So your ribosomes can just basically come up, initiate what's called translation. That's the process of reading one of these messenger RNAs to make a protein based on what it's encoding. And it starts to translate the viral proteins. SARS coronavirus 2 makes two very large, what are called viral polyproteins. They're basically big, long proteins that then get clipped at certain sites to make individual viral proteins. It will make one of these proteins, is called the RDRP, or RNA-dependent RNA polymerase. That's an enzyme that copies the genome and based on its sequence. So it makes what's called a negative sense strand, and this serves as a template to make what are called subgenomic RNAs. Those are basically viral messenger RNAs, as well as make full-length positive sense copies of the genome, which it can then package into these other viral particles. So as it's making all these viral proteins, as it's making all these different viral RNAs, wow, somebody didn't spell check this figure. That should say transcription, and this should say viral, not Braille. Anyways, it makes all these proteins, and then they start to assemble the nucleoprotein complexed with the genome. Meanwhile, all these other proteins go into the endoplasmic reticulum, go into the endoplasmic reticulum, where they begin to decorate the membrane of the endoplasmic reticulum, which is basically a big sheet of membranes. They get trafficked through the Golgi apparatus and sort of start to form these mature virions, or virus particles. Once they kind of put all of this together, it gets transported to the cell surface, where it takes off this envelope layer of cell membrane with it. It gets released out into the environment, and then this can go on to infect another cell, and this whole process repeats itself. One thing that's not pictured here, which I will talk about in a moment, is that some of the viral proteins, and coronaviruses produce many more proteins besides just the nucleocapsid or nucleoprotein membrane, envelope, and spike. There are a number of proteins that actually interact with the host cell's innate immune system that recognizes viral infection and basically shuts down some of the pathways that are important for mounting immune responses to it. And that will become important in terms of talking about some of the immune processes that are important that I'm actually going to talk about right now. So let's talk about basic immunology. Immunology is incredibly complicated, and I think even people who are immunologists, which I'm immunology adjacent as a virologist, but I'm not a hardcore immunologist. I think immunologists find immunology very complicated as well, which is probably why they also find it fascinating, and I find it fascinating certainly for that reason. But I'm going to go over some of the basics because there are a lot of unknowns about immunology. But here's some of the things that we do know that can be very widely applied. First of all, there's really two parts of your immune system. There's the innate immune system, and this can occur within cells, and that's what I was just mentioning. These cellular processes to prevent virus infection is mediated largely by something called the interferon signaling pathway. And there's multiple types of interferons, but the ones we're especially going to talk about today are type 1 and type 3 interferons. And basically what happens is a virus gets into the cell, and as I just showed you, there's all these different steps where there's viral RNA in the cytoplasm. Normally, there's not double-stranded RNA in the cytoplasm, but here, while the virus is replicating its genome and making all these subgenomic RNAs, there is. So one of the ways that the interferon system can get turned on is by recognizing that there's double-stranded RNA here. The cell says, whoa, this looks like a virus infection, and it turns on these interferons. The interferons get secreted to other cells, and they tell those cells, hey guys, there's a virus in town. You better turn on your antiviral defenses as well. And the virus does have ways of circumventing these pathways, but we don't know very much about how it does that for this particular virus. There's also the cellular innate immune system, and these are specialized immune cells that don't recognize specific things the way that B and T cells do, which I'm about to talk about in a moment, but that can recognize certain features that infected cells have. So natural killer cells, for example, can recognize markers that cells will start expressing sometimes in response to interferon that say, hey, like natural killer cell, come kill me, basically, because there's a virus here. And that's one mechanism by which the immune system very effectively gets rid of virus, is by killing infected cells that you can't really go into the cell and get the virus out of it. So a good way to get rid of the virus in that cell is to just kill the cell. And then other cells, at least when you're talking about epithelial cells, they can divide, make new cells, and the wound will heal effectively when you have to kill cells like this through a process called apoptosis, or programmed cell death. So the innate immune system has multiple ways of recognizing, doesn't necessarily know which virus is there, but it knows that there is a virus there, and so it needs to turn on these protective mechanisms. The adaptive immune system is different, and this is what allows us to have immunological memory, because the adaptive immune system says, hey, there's a virus here, and this virus is SARS coronavirus 2. It recognizes pathogens specifically. There's two basic flavors of the adaptive immune system. There are B cells. B cells predominantly express antibodies. And we've all heard probably about what antibodies are. Antibodies are these Y-shaped proteins that are secreted into the blood or serum, which is the liquid component of blood. Sometimes antibodies are called anti-sera for that reason. But they basically bind viruses directly and can render them non-infectious through a process called neutralization. That's why when we're developing vaccines, or when you're looking at the type of immune responses that somebody has who's recovered from SARS coronavirus 2 or COVID-19, you will look at neutralizing antibody titers in their blood and try to figure out how much of a protective antibody response they had. There are also T cells, and this is only showing the CD8 positive or killer T cell, or CTL, cytotoxic T lymphocyte, which is a very non-catchy name. But T cells work a little differently. So CD8 killer T cells will find a cell that's infected, and it knows that it's infected because the cell is doing something called antigen presentation. And so while antibodies can recognize the virus directly, the T cells can only recognize a viral antigen, or thing that the immune system is targeting, based on it's being presented by another molecule that's called either MHC in mice or HLA in humans. And it has to be done in some cases by a specialized type of cell called an antigen presenting cell. So the T cell itself, this is the T cell receptor here, it can't recognize the virus on its own. It has to recognize it by having an MHC or HLA molecule present that thing and say, hey, this is a viral antigen. Do you recognize it? And the T cell says, oh yeah, I recognize it. And then it kills that cell that's infected. There's another type of T cell that's not shown in this diagram called the CD4 or helper T cell. And it basically recognizes antigen the same way. It has to recognize it when presented by a special type of antigen presenting cell called a macrophage or dendritic cell. But its job is not to go out and kill infected cells. Its job is to tell the B cells and the killer T cells that they should start dividing, they should start differentiating, and they should start getting out there and doing their jobs. So the adaptive immune system is incredibly complicated. It's all coordinated by these helper T cells. Both the killer and the B cell responses are absolutely critical for clearing viral infections. So we still don't know that much about what immunity is for this virus long term. We don't know how long the immune response lasts. We don't know how protective it is in everybody. But I'm going to talk a little bit now about some of what we do know. So the first thing I'm going to talk about is what we know about the innate response. And a few studies have come out about this, and full disclosure, this is also what I work on. So I have my own study that's coming out hopefully in the next couple weeks, if I can ever find time to finish my preprint. But this was a great study that was done using samples from patients, using samples from experimentally infected ferrets, and using samples from cells that were infected in cell culture. And it really compared, it used a technique called transcriptomics, which looks at all of the mRNAs being produced by the cell. So it's looking at the host response to infection. And it found, and it compared it to influenza, it found something very interesting. And that is that all pathogenic viruses can suppress type 1 and type 3 interferons to a certain degree. But SARS coronavirus 2, and especially in association with symptomatic COVID-19, appears to do a very, very good job of that. It really reduces these innate cellular antiviral responses that are known. And the result of that is far less secretion of interferon and increased secretion, interestingly, of pro-inflammatory cytokines. And while I don't get very much into pathogenesis in this lecture, we can certainly discuss any questions you have about it because it's still very much an ongoing area of research. It is thought that severe COVID-19 is associated with a phenomenon called the cytokine storm. And that's basically all of these types of inflammatory cytokines. So IL-6, IL-1, there's another one that's not shown here called TNF-alpha. High levels of these cytokines are responsible for immunopathology or inflammatory responses that are essentially unregulated and result in harm to the host tissue. The best known version of this in COVID is the lungs, damage to the lungs that can be done. For both SARS and MERS, as well as now SARS coronavirus 2, we know that patients who are very, very severely ill with severe respiratory disease often develop fibrosis or scar tissue in their lungs, and this can result in permanent lung injury. And this is different than seasonal influenza, which they compared it with. So with seasonal influenza, you also get inflammation, you also get immune cell recruitment, which is the purpose of inflammatory molecules and inflammatory chemokines and cytokines, but it's not to the same degree that it is in severe COVID-19. There's more regulation to these processes. And so this gives us a little bit of a clue that the virus itself is capable of subverting these important innate antiviral responses. Now, in some people who have milder disease, and this should not say that, this should say innate immune responses are mildly suppressed. Inflammation is regulated. I'm sorry about that typo. But normally this happens for all viral infections, but this gives us a bit of a clue. What happens in severe disease, probably in mild infections, patients are having a more effective interferon response and a more regulated inflammatory response. So let's move on to antibody responses. I should add that an important part of the innate immune system and this innate immune signaling is that it does tell B cells and T cells, like, hey, there's a viral infection over here. Maybe you guys should start mobilizing and getting ready to potentially recognize some antigen. So this is a pretty unnecessarily detailed depiction of how antibody neutralization works. But it still, I think, illustrates what's important. So this is the receptor binding domain of spike. It's showing it here binding to ACE2, its receptor. And this is how normally the virus gets in, as I mentioned. These are the antibodies. This is a monoclonal antibody. This depicts a Fab fragment, which is just the business end of a monoclonal antibody. And it's showing how all of these processes can be blocked in order for neutralization to occur. So this is basically showing that here the antibodies binding spike, it's preventing it from binding its receptor. In this case, we're seeing that it's blocking membrane fusion. So another process that has to happen, as I mentioned, is that it has to fuse with the membrane of the endosome in order to get its DNA or its RNA genome out into the cytoplasm so it can start replicating. That requires what's called a 6-helix bundle. It's not important to remember that, but it basically requires this protein structure to make a hole in that membrane so that the genome can go through it. And antibodies can block that process. So this is how antibody neutralization works in a variety of different ways. You can also generate therapeutic monoclonal antibodies that block ACE2 and prevent that from binding to the virus. So there are a number of ways that antibody neutralization can occur, but how often do antibodies get made that are actually effective neutralizing antibodies? Well, most of the time. You may have heard in the news that there's evidence that antibody levels drop in some people or that some people don't have antibody responses. I didn't show it here because there's a million like serology studies out there where people have looked in people who've recovered convalescent SARS coronavirus 2 cases, and most people have antibodies. It looks like at least 90% of people develop some type of antibody. So this is how the antibody response normally works. So you have the virus going up. The virus will peak. At the beginning, you have a type of antibody called IgM. IgM is the first type of antibody that a B cell will make, and it's lower affinity, meaning it can bind more things. It just doesn't bind them as well. So IgM is sort of the first line of defense. Once those CD4 T cells, the helper T cells that I mentioned, get involved, they give the B cell a signal that says basically stop making IgM and start making either IgG or IgA. IgA is a type of antibody that's found in mucosal surfaces, such as the airway, for example, the inside of your nose. IgA is important for what we call mucosal responses on those mucosal surfaces. IgG is what everybody talks about. This is the type of antibody that you measure effectively in blood or serum. IgG and IgA antibodies are more likely to be neutralizing than IgM. So initially, IgM comes up, first line of defense, then IgG and IgA go up. And then over time, after you clear the virus infection, those will go down, but they won't go down all the way. They won't be gone. There are still memory B cells that are capable of making these antibodies. They'll usually just go down to a baseline level. Those memory B cells that make these antibodies are still hanging around as well. So if the virus comes back, if there's another exposure, they will do what's called the anamnestic response, and they start cranking out tons of IgG, tons of IgA in very short order. So that's how vaccines work. That's how the memory immune response works. Now, what does this mean for protective immunity against SARS coronavirus 2? Well, there have been a few reports of reinfection. We don't know how common that is, but this is a depiction of how we think it probably goes for most people in a lot of cases. Within 3 to 4 weeks of infection, you will seroconvert. That means that you just have detectable antibodies, detectable IgG. As shown in the previous slide, your IgM response is relatively short-lived. That goes down below the limit of detection. And then you'll have IgG responses to different degrees. Some people will have a higher IgG response, some people will be lower. Right now, it looks, from the best that we can tell, it looks like disease severity correlates to a certain degree with how much IgG antibody you're producing. So if you have a high response and you go down to a baseline, it might be here where you can still detect it. If you have a low response, it might go down sooner, and maybe that leaves you vulnerable to reinfection. But this is an area that we really don't have very much information in, since the first reinfections have only recently been reliably reported. And so I will answer any questions about this, but for now I'm going to move on so we have time for questions to T cells. So a lot of stuff has been in the news lately about T cells because of a belief by some that people have T cell immunity that already exists against this virus. I'm going to explain that in a moment. What's important is that we don't know if having these T cells means that we are anywhere near so-called herd immunity, which I'm also happy to discuss. We do know that when people are infected with SARS coronavirus 2, they do develop T cell responses. We also know that some people who have been infected with the common cold coronaviruses have T cells that appear to recognize antigens from SARS coronavirus 2. So if you get infected with SARS-2, you get COVID, you will make T cells, both CD4 and CD8 T cells. They'll recognize spike, they'll recognize M and N and other proteins. We also have in some people, some CD4 and CD8 T cells where they've never had SARS-2, but these T cells still recognize at least spike, and in some cases M and other proteins from SARS-2, even though these people have never had it. So what gives? Why do they do that? It's probably because these other common cold coronaviruses do have parts of them that are similar to this coronavirus. So why don't you have antibodies that recognize SARS-2 from these other coronaviruses? That's because the surface of the spike protein is very different from one coronavirus to the next. But remember, I said that T cells have to be activated in response to antigen presentation. There are other antigens from within the spike protein, for example, that may be more similar to the spike proteins from these other common cold coronaviruses, so there's cross-reactivity. Now, all we know, though, at this point is that some people have these cross-reactive T cells. We have no idea how they work. We have no idea if they have any meaningful functional impact on immunity. So that's something that we need to figure out. We certainly can't make any conclusions that they are leading to quote unquote herd immunity against SARS-2. So in general, the take-home message about the immune system here is that it pretty much works like it's supposed to in most people. We develop antibodies for this. We develop T cells. There are these responses. You get infected with SARS-2. The virus will be endocytosed in an antigen-presenting cell. It will be presented. Here's just depicting a dendritic cell, which is one of these antigen-presenting cells, activating CD4 and CD8 T cells. Oops. The CD4 cells then give the B cells an example, a signal to expand. They differentiate into what's called a plasma cell, and they begin secreting antibodies. Similarly, the T cells get activated, they expand, and they go out and start killing infected cells. Some of them start secreting, both B and T cells start secreting cytokines that amplify the immune response, and eventually you'll clear the virus. So that's good news. This is not some magical different virus that's doing all kinds of crazy different things, but there are still a lot of really important big questions about immunity to this virus. And that's how long do these antibodies last? How durable is the protection? We refer to the length of time that you have protective memory immunity as durability. We don't really know much about durability because the only way to find out about it is to kind of wait and see how long people are protected for. Do cross-reactive T cells provide any protection at all? As I mentioned, we don't know. Is naturally acquired immunity as protective as a vaccine? We also definitely don't know that. There's a little bit of data that suggests that vaccines may produce more robust neutralizing antibody responses. I'm happy to talk about that too. And how common is reinfection? Now that we know that it can happen sometimes, how often does it happen and when does it happen? Do people get reinfected anytime, or is it because they have something go wrong with their initial immune response to the infection? We don't know, we need to find out. And then finally, are any of these immune responses harmful, and are any of them harmful long-term? So we know that things like the cytokine storm cause immunopathology or disease that's caused by the immune system, but are any of these memory immune responses harmful? Will it lead to autoimmune or inflammatory disease? Will it lead to any kind of chronic disease? There is some evidence that long COVID is not only just a thing, but probably a more common thing than we initially thought. So how does the immune response play into that? And these are all pretty significant knowledge gaps that we are all actively working on. So with that, I'm going to switch to a question, the question portion of this. And also here is my Twitter and email address. If you have any questions that you think of once this is over, please don't hesitate to drop me a line.
Thank you so much, Angie. That was a fantastic talk. I think we all learned a ton. I'm sure there are a lot of questions. I just want to remind the audience that if you have questions, you can look at the bottom of your Zoom screen and you can either click on the Q&A button and you can write your question in the Q&A box and we will read it to Angela, or you can raise your hand and we will call on you. So while the questions are starting to come in, I will turn it over to Sarada for a moment.
Wow, Angie, that was a very clear explanation on some basic concepts of immunology and virology in general. I have learned a lot, and I'm sure the audience did as well, because we can see a lot of questions coming. So let's get on to them. Frank, would you like to raise your hand so we can let you ask your question, or would you like us to ask your question for you? I'm just going to see if I can find.
Go ahead, Frank. If you're there, go ahead and ask your question.
Hello, can you hear me?
Yes, we
I
can.
can.
Okay. I was curious if people are getting reinfected, what does that mean for the prospects of a vaccine actually being effective?
That's an excellent question, and it's one that the short answer is I don't know. But there are some things about the vaccine trials so far that have happened that suggest that maybe it's not going to be such a big problem for vaccines. So the few cases of reinfection that have been reported, none of them have really come along with good data about the antibody response. There was the case in Hong Kong that suggested that the person who was reinfected, who had very mild or asymptomatic disease actually on the second time around, may not have developed antibodies after the first infection. And so that provides a pretty clear hypothesis for why he got reinfected. In the other cases, there wasn't a serum sample available from their first infection to see if they had antibodies or not. So it's really difficult to say that, they just didn't have a robust immune response the first time around. And so that's why they were susceptible to a second round of infection. We also don't know if it's very common or not. So if it's not very common, then that probably isn't going to make a big impact on vaccines. But there's a couple other reasons why reinfection may not be such an issue with vaccines. And one of those reasons is that from the phase 1 trials and from the animal studies that have been done prior to the vaccines coming out, it looks like vaccines induce really high levels of neutralizing antibody along the higher end of convalescent patients. So, there's this large range of patients. Some develop no antibody response or no detectable antibody response. Some have very high titers of antibodies. All of the vaccine recipients appear to have higher titers of antibodies, of neutralizing antibodies also, which bodes well for protection against reinfection. The other issue is that because vaccines aren't an infection with SARS coronavirus 2, there's none of the virus doesn't have the ability to manipulate the immune system the way that natural infection can. And another study that came out looking at autopsy patients' lymph nodes showed that people who died of COVID-19 often didn't have what are called germinal centers in their lymph nodes. And those are areas where the CD4 helper cells like to hang out and give other cells of the immune system signals to differentiate, to go out and do their functions, and then ultimately to become memory cells, which suggests that maybe in some people, at least with severe disease, their immune systems aren't mounting the type of responses that would allow them to have robust protective immunity for a long period of time after they've recovered. Now, we don't know that in people who have milder disease. Of course, that one study was done in autopsy samples of people who died. So we don't really know if that applies to everybody who gets it. But it does suggest that maybe there are some differences between getting immunized by a vaccine and getting immunized by natural infection that would affect your long-term susceptibility. So like I said, the short answer is we don't know. We need to do a lot more, but there's hope that a vaccine would potentially induce more robust protective immunity than natural infection would.
Thanks, Angie. We have another great question from an audience member, Hugh Barrett. Hugh, would you like to ask your question?
Yeah, can you hear me?
Yes, please. Yes. Go ahead.
So I was just curious, there's a lot of people who never get colds, never get the flu. Are they more likely not to get infected with coronavirus? Is there any correlation between the two, or do they just have relatively strong immune systems?
That's a really tough question to answer. The reason being that there are a lot of variables why some people might not get severe colds and flus. Some of that might have to do with behavior and lifestyle, like maybe they're just at a lower risk of getting them because they're not out in public. Maybe they do a job where they're not exposed to as many people who might be infected. Maybe they practice better hand hygiene. Maybe they're living in a rural area where they're going to be around fewer people in general than somebody who takes a crowded subway to work every day. There's a lot of reasons why somebody might not get colds or flus. There's also cross-protective immunity against a variety of other viral pathogens. So colds in particular are caused by hundreds of different viruses. Many of those are related. The rhinoviruses, for example, I've always had a soft spot for because I did my PhD on rhinovirus. But there's hundreds of rhinoviruses, and there may be some cross-protective immunity against, rhinovirus 39 if you get infected with rhinovirus 16 or 20 or whatever. So if people have been exposed to other viruses and they have long-term immunity, there may be some cross-reactivity, and that might make disease less severe if they get infected again. The other thing is, and this is based on not just my work with Ebola, but other people who work with a similar model. One of the things that I do, I study the host response and its role in disease severity. People have shown this for influenza. They've shown it for SARS Classic. I've shown it with Ebola, that when you use a bunch of mice that are genetically different from one another, you infect them with the same virus at the same dose, they can have radically different disease presentations. And it can be very different in severity. For example, in this model with Ebola, 30% of the animals die from Ebola hemorrhagic fever, basically. They have hemorrhagic disease, it's very severe, it's lethal. About a third of the other mice die, but they don't have that hemorrhagic disease. And about a third of the mice live. They lose a little weight and they get better and they're fine. So that's with Ebola. That's not with the seasonal flu or a common cold. So people genetically and probably epigenetically as well, and this is probably also determined by people's own medical status. Do they have asthma? Do they have other pre-existing conditions that would predispose them to having more severe colds or flus? Those kinds of things can also play a role. So there's a lot of variables, and it makes it really hard to answer that question. But from a basic standpoint, and that may be why we're seeing so many people with these very diverse set of presentations for COVID-19. There are some people who are asymptomatic, and there are some people who are on a ventilator. And there are some people who seem to have neurological disease. There are some people who seem to have gastrointestinal disease. There's a lot of different manifestations, and probably all these variables are contributing to that. But whether or not you get severe colds or flus, there's only 4 coronaviruses out of these hundreds of common cold viruses. So it's hard to say with this particular coronavirus. Now, in Asia, East Asia, it may be a little different. If somebody had SARS coronavirus classic, they may have more protection against this particular virus, because it has been shown that antibodies to SARS classic will protect, at least in experimental systems, they will protect cells from being infected with SARS coronavirus 2. But here in North America, where there have been very few cases of SARS classic in 2003 to 2004, there's probably not a lot of that pre-existing immunity. So I would think that it's not going to necessarily be determined by a general susceptibility to colds and flus. Everybody's susceptible to this, and people will probably develop different severity disease for a variety of reasons. That was a really long way of saying I don't know.
Interesting. Thank you, Angie. We have a question from Eric Wagner. Would you like to ask your question, Eric? Eric? Okay, I'll read out his question. Eric wants to know, do asymptomatic people or those with relatively shorter syndromes have fewer or different kinds of antibody generated in response?
Yeah, so we do know that. It does appear that people with asymptomatic disease have lower antibody titers, and that those antibodies may not be detectable for as long. What we don't know is how that impacts long-term protective immunity. So as I mentioned briefly, with the memory immune response, but specifically with the antibody response, there is this anamnestic response that's been characterized. So you may not have enough antibodies circulating in your blood to be detectable by a laboratory test, but you may still have those memory B cells that are there just waiting to respond. And if you do, then they will start churning out a lot of antibody very quickly. We don't really know what that response is like in people with asymptomatic or very mild disease, but we do know that in general, asymptomatic or very mild disease does result in usually weaker antibody responses. But the significance for functional immunity and long-term protective immunity isn't currently known.
Thanks, Angie. We have a question about convalescent plasma from Aaron Beckman. Aaron, are you there? Aaron, are you there? You want to ask your question? That's okay. I will ask it on Aaron's behalf. Aaron wants to know, has research shown any positive results in the use of convalescent plasma to treat COVID patients?
Oh boy. Yeah, this question is kind of a doozy just because this whole topic has been very confused by the way that the FDA issued an emergency use authorization for convalescent plasma. That was an unusual circumstance because the data that was used to justify that EUA was basically observational, meaning that there were not really robust controls. It was not a randomized controlled trial that you would normally rely upon to say for sure if something works or not. So we know from previous work that convalescent plasma in some cases does have some kind of clinical benefit. So it's not out of the question that it does. There are studies going on to see that are randomized controlled trials to see how significant that benefit is. But right now it's very difficult to put a number to how effective convalescent plasma is. And that's primarily just because those trials weren't going on or those trials were not complete when the FDA approved it. So the jury's kind of out. My personal feeling, which is, it's my opinion, it's not a completely science-based opinion, is that it probably has some impact. It probably also, though, isn't the breakthrough that some people have made it out to be.
All right. Thank you, Angie. We now have a very interesting question from Karen Dell, and I think I'll join her in asking this question. Karen, would you like to ask your question, please? Karen, are you there? Karen, could you unmute yourself? Yeah.
Hello. Yes, my question is about cytokine storms. You mentioned it, and I've read about it in the literature and things too, but I'm just wondering for some of the maybe non-scientists who are in the audience, if you could explain what happens during a cytokine storm and how that would impact the physiology or pathology of COVID-19. Sure,
yeah, and that's a great question. And it's one that so I will back up and say that cytokine storm can mean a lot of things. It's used generally to describe sort of an uncontrolled release of inflammatory cytokines. And maybe I should explain what cytokines are. They're signaling molecules like interferons that are secreted by cells that are either infected or that are given a signal to start secreting cytokines. And normally the inflammatory response is really, the cytokines drive it. And it's really important for containing an infection because what inflammatory cytokines do is they basically make it possible for the cells of the innate immune system and antigen presenting cells like macrophages particularly to migrate to a place where there is an infection, maybe start killing some cells, also pick up some antigen, take it back to the lymph nodes to start activating B and T cells. It's basically a way of saying, hey, infection is here. It's in the lungs. It's not systemic. You need to come here to this place where the cells are actually infected. You need to start killing these cells. You need to start gobbling up the virus. You need to start containing the infection. What's critical is that those inflammatory responses are turned off once the infection is cleared. In some people, for reasons that are not clear, those inflammatory responses, those cytokines, continue to be produced, and they're not produced in a way that's tightly regulated. So these cells just start dumping out these inflammatory cytokines, and they can dump them out into the bloodstream, which in the lungs, if they're doing that, that can create a general inflammatory state. It can also recruit all of these pro-inflammatory cells to the lungs. And if there's no more virus there, or if there's not that much virus there, if it can be controlled through other means, those cells will actually cause tissue damage or immunopathology. So a cytokine storm is kind of a general phrase for when those inflammatory responses become unregulated and start recruiting all of those inflammatory cells to one site or not. They can be systemic as well. So in Ebola virus disease, it's thought that the bleeding and the hemorrhage that occurs is because of a systemic cytokine storm. It's not because the virus is like killing the insides of the blood vessels. What we don't really understand very well for COVID-19 is what this cytokine storm looks like across the board. We know some of the basic inflammatory cytokines that are involved, but we don't know if, let's say, one person has very severe lung disease, another person maybe doesn't have as severe of lung disease, but they're having some of these vascular complications or the cardiac complications. We don't know if that's the same kind of cytokine storm, if it's the same cytokines being expressed, if they're attracting the same types of inflammatory cells. Because even though there are macrophages, there are dendritic cells, there are all these different kinds of sort of innate immune cells that are involved in this, there are a bunch of different kinds of those cells that all do different things. Like some macrophages are very into promoting inflammation. Some are involved in wound healing, things like that. So if it's recruiting, we don't know much about whether it's recruiting certain kinds of cells to these sites. We don't know if it's certain cytokines that are causing it. We don't know what causes the loss of regulation. It probably has something to do with the suppression of type 1 and type 3 interferons. But the bottom line is this is still a very active area of research, one that I'm involved in, as I mentioned before. And based on my own work, there's not really any clear answers right now. I think we're going to need to do a lot more in patients to try to tease out which are the key players in mediating specific types of COVID-19 cytokine storms.
Thanks, Angie. We have another great question from one of our audience members, Bob T. Hey, Bob, are you there? Would you like to ask your question?
Sure. So, as I understand it, there are two types of viruses or coronaviruses that initially were detected, one coming from Asia and the other one from Italy, that were slightly different. Wanted to understand the impacts of the variants and how they're different, as well as the fact that I thought I saw something that said that there's been further mutations of the virus. So could you discuss that and the impact on what that means in terms of producing vaccines, whether you might have to have more than one type of vaccine?
Yeah, absolutely. And that's a fantastic question. This was just in the news again yesterday. So before I answer it, I'm just going to say one thing that I probably should have said when I was going over the virology portion of my talk. And that is that one thing about RNA viruses is that they have a much higher mutation rate. The enzyme that copies the genome lacks one thing that the enzymes that copy DNA genomes has, and that's the ability to proofread. So it's basically making a copy based on the template, but if it makes a typo, essentially it can't fix it. Now, that's for all RNA viruses, so they all mutate much more quickly than DNA viruses or than we do, because they don't have that proofreading ability. One thing that is unique to coronaviruses, though, is that they have another protein called an exonuclease that is capable of some proofreading. So while their mutation rate is higher than ours, or higher than a DNA virus, like a herpes virus, it's lower than for something like influenza. So these viruses do, when they replicate, they make mistakes, they make mutants. That's normal for viruses, for RNA viruses. Every time they replicate, there may be one or two mistakes in the genome. Some of those mistakes will have no effect. There will be no evolutionary selection to keep them around. Some of them, it might just make that mistake more frequently, still has no effect. So there's something called founder effects, in which those genomes will stick around. They may be observed as variants, but they don't necessarily do anything. And then occasionally that mutation will happen in a place that gives the virus some sort of advantage. And then it will be under what we call positive evolutionary selection. And that variant will persist. Just because, though, it gives the virus some kind of advantage in terms of replication doesn't mean that it's necessarily going to have an effect on transmission or on pathogenicity. So we've sequenced thousands of different variants, and there are a number of different variants besides just two. I think what you might be talking about is the so-called D614G mutant. These are not different strains of the virus because none of them are immunologically distinct from what we can tell. But this D614G, or as one of my colleagues calls it, Doug, that means that basically there was a mutation in the spike protein that resulted in an aspartic acid at position 614 being replaced with a glycine. These are the amino acids that are the building blocks of the protein. It's not, though, in a part of the spike protein that is thought to be important for antibody neutralization. The spike protein is actually 3 spike proteins put together, and this mutation occurs in the interface between those, but it's not where spike actually binds the receptor for the virus. So it probably doesn't have much of an effect on the virus actually getting into cells. Now, it's been reported because the G variant of this is more common in North America and Europe, that it has some sort of selective advantage. So it's under positive evolutionary selection. And it's been proposed that this is because the virus is more transmissible. It's very unclear whether that's true or not. And this was a preprint that was released yesterday that showed that the G mutation, the G variant of this, was completely dominant in the wave that was in Houston over the summer. Now that may be, but it also could be founder effects. It's quite clear from looking at the sequence data that early on, the G variant was present in North America, and that it may just be that that virus got more of a foothold to begin with. And so that's now become the dominant variant in the US. It doesn't necessarily mean that it's more contagious. Some people have done some studies in cell culture to show that it may provide what's called a fitness advantage. It may make the virus more capable of infecting cells or making more virus in those cells that are infected. But those are cell culture experiments. They're cells in a dish, and that's very different from a complex human being. And so what the bottom line is that it's definitely worth looking into to see if that variant does increase transmissibility, for example, in people. But based on the data we have so far, just because it's more prevalent doesn't necessarily mean that it's actually more contagious. And this is a very active area of research, and it has been very controversial. But the good news is we can't rule out the possibility that a variant will emerge that will be in a critical part of the virus. So the receptor binding domain of spike, for example, is thought to be very important for antibody neutralization. If there are any mutations there, it's entirely possible that it could make a vaccine less effective. So that's why even after we have a vaccine that's safe and that works, we need to continue doing surveillance to make sure that we're not seeing variants emerging that might have an effect or might allow the virus to escape existing immunity by a vaccine. The good news is, though, that many of the vaccine platforms that are leading the race, I hesitate to call it that because I think there's going to be multiple winners to this race, but all of the vaccines that are currently in phase 3 clinical trials are all very easy to modify because they're basically made through genetic engineering, and it's very easy to insert small changes like these types of mutations into those vectors. So once we know that these vaccines are safe and they work, adapting them to any variants that should emerge should be fairly straightforward and easy to do. And that's good news. The fact that coronaviruses have a lower mutation rate than other RNA viruses means that we're probably gonna still have to be vigilant, but this is gonna happen less often than it does for something like influenza virus.
All right. Thank you, Angie. We now have a couple questions on herd immunity, one from Sarah Moorhead and one from the audience. So basically, what constitutes herd immunity? And in your opinion, how long would herd immunity potentially last?
So that's a great question too. And I have to say, during this pandemic, herd immunity has been one of the most misunderstood concepts in terms of the public discussion about it. So before I kind of go into that, I'll answer the question, how many people have to be immune before we have herd immunity first? And the correct answer, I think for everybody is, I don't know. It can differ for every virus. It depends on how robust the immunity is. It depends on how you're generating that immunity. But I should say that herd immunity is the concept that all viruses, being obligate parasites, they all need a host. And if you have enough people in a given population, the herd, who are completely resistant to being infected, then that virus will have nowhere to go, and it'll eventually die out and not be circulating in that population anymore. For every virus, that will be different. As I mentioned, it depends on a lot of different variables, including how effective a vaccine is. I think one thing that has also been misunderstood is that we've never achieved herd immunity for a virus through natural infection. We've always achieved herd immunity through vaccination. And in our previous experience with vaccines, in order to get herd immunity, we need probably a minimum of 60% of people to be vaccinated and immunized and resistant to infection. It may be different. Maybe those T cells that I mentioned before will have a function, and we can get away with fewer people being vaccinated. Maybe, we will have some level of virus circulating in the population because maybe the vaccines will only be partially protective. It's really hard to say, but probably we need a minimum of 60 people to get vaccinated to achieve herd immunity and really not be living with this infection anymore. But we know from really all the vaccines we've developed in the past that we can, with enough people taking those vaccines, we can effectively eliminate viruses from our population. We've done that effectively in most of the world so far with polio. We've eradicated 2 viruses, smallpox and rinderpest virus, which is an agricultural pathogen. And we can do it potentially. We may not be able to eradicate every virus, but we can definitely reduce it so that we are only dealing with at most sporadic outbreaks of a given virus, depending on how well the vaccines work and how many people are willing to take them. So I think we're still a long way from herd immunity, but once we get a vaccine out, once we're able to distribute it widely, then the question is going to be how many people are willing to take it, how many people are going to get vaccinated and put concerns about the vaccine aside. And that's where it's on scientists like myself to communicate to people, that these vaccines will be safe and effective, that they've been approved. We need to talk about how those vaccines work and sort of win hearts and minds to get people on board with vaccination. That's how we're going to achieve herd immunity.
Thanks, Angie. We have a related question about immunity from Stefan Solfeld. Stefan, are you there? Would you like to ask your question?
Oh, yeah, sure. I can do that. Thank you. So as an honest scientist, you say correctly that we do not know for how long people will be immune to reinfection. So this is a very general statement. But as much as I know, and I don't know much about this, there are a bunch of ongoing studies measuring antibody titers in previously infected patients. And in these studies, nobody has been found to be reinfected within the study time frame, which by now covers about 3 to 4 months. Some anecdotal examples have been reported outside of those studies, but not within these studies, at least to my knowledge. Wouldn't it be better then to say that infection very likely protects against reinfection for at least 3 to 4 months to our current knowledge, and probably longer? And what is your hunch for how long this is probably the case based on other related viruses?
Yeah, so that's a great point. And so we have, the cases of reinfection are somewhat anecdotal, but there have been several cases now that are proven cases of reinfection where the person was infected with one virus, they sequenced the virus, the second time around they were infected with a completely different virus that there's no way it could have been the same virus. So that's clearly reinfection. What we don't know, and you're correct, absolutely, nobody in any of these long-term longitudinal serology studies has been reinfected. Which is great. And to me, that suggests that while reinfection can occur, and you do expect to see rare events happen when you have a lot of people infected with this virus. So if it's something that's a 1 in a million event, we've had millions of people get infected with SARS coronavirus 2, it's not terribly surprising that we'd find a couple cases of reinfection. But you're absolutely right that these long-term serology studies show that it's probably not very common if they're not detecting anybody within these studies that are getting reinfected. Now that said, I always try to be cautious in balancing like what we don't know with what probably is the case. Now, I think probably reinfection is probably uncommon. I think it's probably rare, and I think for most people, you know, my PhD advisor said something that I thought was really true, and he's like, a lot of people think that because this pandemic has upended all of our lives so much, that people are suddenly attributing like these magical properties to this virus that we don't see for other pathogens, despite the fact that many of them are other virologists who know better. That why should this virus be any different than any other virus in general? Yeah, in most people, it induces immune responses. We can't say for sure that those immune responses are protective, and we can't say how long they last, but it looks like this virus it's pretty normal as a virus in most people. It's not because it's some exceptional virus that it has become a pandemic. It's become a pandemic because the policies and the responses to the virus were inadequate to control its spread around the world. So I think you make a great point that perhaps it is better to say that infection does very likely protect against reinfection for the vast majority of people for at least a few months. And I would expect that protection to be longer too. I think there are a couple problems here that one of those issues is coronavirus research itself and the state of that research. We have some evidence that the common cold coronaviruses can reinfect people. It's very unclear how often that happens, because think about when you get a cold, very rarely are you going to the doctor and getting a diagnostic test to find out exactly which virus you're infected with. People have not looked at long-term immunity for many of those common cold viruses just because there's not always funding for it. And coronavirus funding in particular operates on a very boom and bust cycle. So SARS Classic emerged, lots of money. SARS Classic doesn't come back, money goes away. MERS emerges, more coronavirus money. MERS doesn't appear to be very transmissible from person to person, money goes away. So we have these large gaps in our knowledge in general about long-term immunity to coronaviruses. We've never managed to make a vaccine for any of these coronaviruses because SARS, there was nobody to do a phase 3 clinical trial, and after the initial outbreak was effectively controlled, and it hasn't reemerged. MERS, as I mentioned, isn't very transmissible from person to person. So we don't really know that much about long-term immunity, and people are making, I think, a lot of assumptions based on the few studies that have been done either with animal coronaviruses or with the common cold coronaviruses, and it may be different for this virus. We can't say how long protection against reinfection is going to last for this, but I think that it's certainly likely that that protection for most people will probably last a year or longer. We just don't really know, and we'll have to find out. But I think for most people, you're absolutely right. They do develop protective immunity that protects them for reinfection, at least for the length of time that we've been looking at this so far.
All right. Thank you, Angie. And now we have a question from Carl Zabura. Carl, are you here? Would you like to ask your question?
Hi, this is actually Carl's wife. So I have a teen son, and his close friend had COVID. This was probably a month ago, or 2 months ago, maybe. Now my son feels he can spend time with this boy because he has the antibodies, and he can't infect him. So my question is, what should I tell him if he wants to hang out with him without masking? This is like, so the boy had it 2 months ago, 2 weeks ago, 2 weeks after the boy had it, or actually maybe a month after the boy had it, he says, oh, well, the boy has antibodies. I don't need to mask. I can just hang out with him because I won't get it from him.
Yeah, I think that's not an assumption you should make. Chances are he probably does have antibodies, and it's probably okay. But at the same time, one thing we do know from some of the vaccine studies is that, and this is where the reinfection thing gets complicated, because we know that the problem with vaccines against respiratory viruses, is they don't offer complete protection against mucosal infection. And what we've seen from some of the non-human primate studies that the vaccines have been tested in is that some of them, even though they have high levels of antibodies induced by the vaccine, when they're challenged or deliberately infected after being immunized, some of them have evidence of subgenomic RNAs in their nasal swabs. That suggests that there's replicating virus in there, which suggests that vaccine may offer protection against disease severity, but it doesn't necessarily offer complete protection against infection. And while that may not be an issue, it's still obviously a huge public health benefit if people are not getting sick, if they get infected, but it may mean that they can still transmit it to others. And because we don't know enough about that, we don't know enough about mucosal neutralizing immunity, I think that until we know more about that, I don't think it's safe for people to assume that because they have antibodies, they can run around without masks themselves in public. I have not had COVID and I'm seronegative, but if I was, I would still wear a mask in public. I would still wear a mask when hanging out with people. I don't hang out much in person with people, but I think that it's an oversimplification of what we understand about protective immunity right now. That even if he's protected from severe disease in the future, he may not be completely protected from infection. And may be able to transmit the virus to others were he re-exposed. So I think that you should tell your son that no matter what, he should be wearing a mask when dealing with people outside of his household, as much for his own protection as for just being a courteous practicing good respiratory etiquette and being a courteous fellow citizen.
Of course, I think your son had COVID.
Well, thank you, Angie. And of course, there are many, many more questions, and we would like to continue discussing with you for much longer, but I guess we are almost 30 past the hour, so we probably will have to close at this point.
I just want to say thank you so much, Angie. That was a fantastic talk. I think we all learned a ton, not only from your talk, but also from the questions after your talk. I also want to thank the audience for joining us tonight. And I want to remind everyone to please come back next Wednesday, October 1st at 7 p.m. to hear about the epidemiology and the transmission of COVID-19, which includes how and why the disease affects certain populations of people and how it spreads. And again, on October 7th, to hear about ongoing efforts in therapies and vaccines. And I want to remind everybody that you will again have another opportunity to get all of your questions answered with Angie if you did not have a chance to do that tonight. And thank you all for joining us, and we hope to see you next week. Have a good night.
Thank you, everybody. Thank you, Angie. And yes, please do join us next week.
Thank you so much. I'm looking forward to it.